Thank you, Marika. Do you see me and hear me fine?
Yes, we see the slides and we also hear you.
Excellent. And thanks for the good words. I must start by saying that this is a fairly new area for me. And of course, it's thanks to the work of giants who've come before that we've been able to make fairly good progress recently, and hopefully even more in the next few months. I want to tell you about maybe more generally the research we are doing at MILA in the space of drug discovery, but in particular about our project on drug combinations for antivirals for COVID-19, which we call RECOVER, that is funded by the Gates Foundation and in collaboration with Relation Therapeutics, UCL, Imperial College London, sorry, and Juvenescence and Scripps Research. So let me move, yes. So let me say a few words about the work we're doing in molecule discovery at MILA. So what motivates researchers in AI and particularly deep learning to work in this area? Two things. One has to do with the social impact. So I'm going to be talking about drug discovery, but actually a lot of the techniques we're working on, we've been also exploring their use in the discovery of new materials for fighting climate change, designing new batteries or new materials for carbon capture. And I really think that these AI techniques can make a crucial difference in both of these areas to initiate a phase transition in the way that new materials and new molecules in general are being discovered. The other reason why researchers, PhD students at MILA are excited about these areas is because they are not only impactful, but involve really challenging questions in basic research in machine learning at the frontier of our exploration to improve deep learning and approach eventually human level AI. So let me mention a few of these, and during my presentation I'll go a little bit in some of these things. So one of them, which is maybe less obvious, is that the active learning aspect that we are exploring in drug discovery is really connected to fundamental questions in how an agent can act in order to improve its ability to acquire knowledge efficiently. So, Bayesian optimization, of course, is now well established. The deep learning versions of this are a bit more recent, and this is the kind of thing we've been doing in the context of drug discovery. But it really asks basic questions about what are the right objective functions, what are the right training frameworks in order to enable a learner to decide where to explore, really. Okay, so it's an exploration question. And of course, the exploration question is something that has been in the mind of researchers in reinforcement learning, which is one of the big areas of research at MILA and in general in the world in machine learning. And where it comes in addition to the exploration question in the case of drug discovery, is the question of exploring a sequence of changes. So typically, not for drug combination, but in the case of de novo drug discovery, we are asking the question of how could I change a molecule by adding or removing pieces in order to achieve some goal. So that's another reason for exploring these topics. Another one, which is really hot, is the topic of out of distribution generalization. So what is that? Classical work in learning theory has been always assuming that the test data, the field application of the trained learner would be examples coming from the same distribution as the training data. But in many realistic scenarios, this is not what happens. You train from some distribution and you're going to be deploying in a different one. Now, in the context where you have a learner which is exploring, it's even more the case, because if you think about just discovering new molecules or new combinations of drugs, as we continue that exploration, we are essentially pushing the boundary of things we have explored pushing outside of the realm of the things we know. And so in a way, we're systematically going out of distribution. So that's an interesting kind of problem. And again, it connects to a lot of research questions, basic research questions that are of interest for researchers in machine learning. Another one, which is more basic in some senses, we are dealing with graphs. We're dealing with molecules. We're dealing with very structured data sets, as I will talk about, for drug combinations. And so these graph neural nets and the representations that are learned on them is a very current topic that goes, of course, well beyond the world of molecules, for example, in social networks. And then the last topic, which hopefully I'll have a bit of time to talk about at the end, is the connection to causality. I think in biology, in particular, in drug discovery, really what we would like to use machine learning for isn't just to capture statistical dependencies and to search over them, but it's also to capture causal dependencies. We want to make sure that the actions of, say, a drug or a couple of drugs or a triplet of drug is going to have the effects we want, and especially potentially in a different setup than the one we are getting data for. So somehow making progress in getting closer to the causal understanding, which obviously is a very big challenge, is also touching on some of the very current research questions that we are interested in at MILA. There's a long tradition of research in causality in science, in economics, in social science, and statistics. But it's only fairly recently that machine learning researchers have started to ask questions about how to connect what they were doing with causality questions, and maybe how to ask new questions, for example, about learning the right representation space that corresponds to causal variables. All right, that was a long introduction, but I wanted to make sure that you get a big picture of like why we are into these questions. Now, going a little bit one level down, we have two current projects at MILA regarding antivirals for COVID-19. One, which I'll spend more time on, which is about combining existing drugs, and the other is about creating new molecules. So the advantage, of course, I mean, for this audience, I don't need to motivate the first project of combining existing drugs. For the de novo molecules, the challenge is, of course, now we're looking at a much larger space. It's also an opportunity. And this is also a place where, as I mentioned earlier, reinforcement learning becomes a crucial tool. Yeah, so drug repurposing is obviously something important here. Hopefully, we can get to treatments faster because we are going to be evaluating drugs that have already been tested in some way. And of course, the challenge is the number of possibilities that we need to consider, and there's no way we can assay all of them. In our project, the Recover project funded by the Bill and Melinda Gates Foundation, we start from the amazing resource of the Reframe library, which comprises 12,000 molecules that could be repurposed in this context. So assaying those molecules has cost $50 million, and the work that has been done is going to be a starting point for our work on combinations. So let me move on. Why drug combinations? Again, for this audience, I don't need to spend a lot of time, but it's important, and it's something we still not completely sure what's the right way of dealing, of measuring, and that is a question of synergy. So In a way, we want to pick drugs that are useful individually, but also somehow when they are put together, create a stronger inhibition than you would otherwise get. Presumably because they maybe target different pathways, and they make the effect more robust for reasons that we'd like to understand, but that we care to measure. So there are actually many measures that were proposed for estimating the synergy between a pair of drugs. Unfortunately, when we look at the different measures, they are not always very well correlated with each other. And so I think this is something that hopefully our work will help provide some answers to. There's a question of the size of the space. So with 12,000 molecules, that's 72 million pairs. Even if we were able to assay 5,000 of them per week, it would take 277 years of search. So this is absolutely not feasible. And so we'd like to fill this matrix of pairs of drugs, but we'll only be able to ask questions about a very small subset of those pairs. And in a way, we want to generalize to the other combinations, at least to find the ones that are synergistic. And as I mentioned earlier, this whole research area is heavily influenced by the thinking about exploration versus exploitation. And the reason this is possible is because the way we're going to be using machine learning here isn't that magically AI is going to come up with the right combination of drugs, and then we're going to assay them and then move on to mice and then to humans. This is not how it's going to work. We're going to have several iterations of assays. We're going to do something like be able to send queries every week, starting hopefully in January. And so when you're in this situation where you're able to do to query, think of this like an oracle, there are assays which can only give us very few answers. There is the logical thing to do, the optimal thing to do is not to go directly for your current best guess. The optimal thing to do is, of course, to include your current best guesses, but also to include other queries, other candidate combinations that somehow will give new information to the learner, so that at the next iteration and the future iterations, you'll be able to do better than if you hadn't done that exploration. So exploitation means, go right away for the things you think are good, but there may be good things elsewhere. And so depending on your estimated uncertainty in these other places, you might want to do quite a bit of exploration, actually. And our experiments that I'll talk about confirm that in the case of searching in the space of drug combinations. So we're going to have this loop, with testing of new combinations of compounds on one hand, and on the other hand, a machine learning pipeline, a very heavy machine learning pipeline, which produces recommendations, and the results come back, and this goes on. And the plan is for this to go on for something like 8 months. So quite a bit of iterations. Of course, I'm not a biologist or a biochemist, and so I'm going to focus on the machine learning aspect of things. And please excuse my poor use of the jargon in the pharmaceutical world. All right, so let's focus a bit on this recovery project. The machine learning pipeline has essentially these 3 building blocks. First of all, we're going to have data on many things that are going to feed the learning machinery. We have known synergies, and of course, as we do more acquisition, more assays, we're going to get more of these. And then we're going to have this library of drugs for which we know something about the structure. We may have fingerprints. We also may know information about targets and the corresponding genes. We already are using protein-protein interactions data, and we'll be able to measure differential gene expression as information that can give us clues about which combinations of drugs actually are changing the right things in the cell. So that's sort of on the input side. In terms of the machine learning machinery for making predictions, this relies heavily on graph neural nets, and I'll go in much more detail of that. The output of these graph neural nets will be these predictions for pairs of drugs in terms of synergy, and as well as an estimated uncertainty around those predictions. And then finally, the last block is about the exploration versus exploitation challenge that I've been talking about. We are using active learning methods that are exploiting a notion of acquisition function. In other words, we're combining the predicted synergy with the predicted uncertainty about the synergy to score different potential candidates. And that scoring function is called an acquisition function. And this is how we decide which combinations are going to be tested for the next iteration. All right, so this is a slightly more detailed way of the big picture here. There's this loop with assays in the bottom and machine learning on top. The starting point here, if you want, is the knowledge graph that'll go into more detail that captures essentially all of the data we have. So instead of thinking of a data set, have that data set is structured as a graph. And then, as I mentioned, we have the whole active learning framework that is going to produce the suggestions. Initially, the suggestions are going to go to primary assays that are going to evaluate inhibition and look at combinations of 3 levels of concentration for each of the 2 drugs. So we're creating a 3x3 matrix of outputs that are going to eventually feed the knowledge graph. We also are going to have secondary assays where we get information about expression, which in our preliminary experiments suggest that is a very strong clue that can help the machine learning prediction. And of course, those drugs that are looking good on both will then be passed on to mice assays that are going to happen in parallel to all this during the whole time of the project. So we have started testing many of the building blocks from a machine learning perspective, since we don't have the COVID-19 assay results yet. We've been doing tests on the cancer cells inhibition data set that has already shown to that combinations can be useful. And so one of the first things to do is to verify that indeed using the here just the information about the structure of the 2 drugs, we can do a fairly good job of predicting the growth inhibition associated with pairs for which there is data, of course. In this case, we can't query new combinations that are not in that data set. But that data set has 8 million data points. All of these are pairs of drugs over 2,000 cell lines. Now, the other thing we have tested, which is, of course, important for us, is the effect of using active learning versus doing sort of brute force, random choice of pairs that would take much more time to discover the good combinations. So what we see in the graphs are different runs of our active learning setup, where each iteration, there are 20 iterations here, each iteration involves asking for 100 new data points. So in the brute force screening, those 100 are chosen randomly among all the tested pairs that exist in the data set. And in the active learning setting, the pairs that are selected based on what the current acquisition function scoring gives. And so what you see, and then on the y-axis, it's the on the left, it's the proportion of combinations that are in the top 1% that have been discovered. So you want this to be high. And in the second graph on the right is the cell growth that we're able to inhibit using the best combination found up to now as we continue that process. So clearly these active learning methods are powerful in this context. And in this case, we use a particular acquisition function that's called UCB that I'll talk about next. The computations needed to do these experiments is fairly substantial. For each of these updates, when we get a slightly modified data set, we need to retrain or fine-tune, but the whole existing data, and redo also the hyperparameter search, or at least verify that the hyperparameter search it continues to be good. And so there's a whole infrastructure for doing these things. Let me go back a little bit more detail in the data. So we're using a large resource on protein-protein interactions from different sources. We're using Morgan fingerprints to represent the drug structure. We could also be using graph neural nets, and we're actually using graph neural nets in another project. This is not something we've done yet, but this is something, I mean, it's a bit more expensive, so we haven't done it yet, but it's something we could do. We, of course, are using information about the drugs and the corresponding targets, and about gene expression using the CMAP L1000 data set. So graph neural nets. Graph neural nets have been around already for a few years. They're being used more and more in many applications. Of course, in areas like social networks, natural language, knowledge representation, knowledge bases, knowledge graphs, and so on. But in the world of biology and drug discovery, there's already a few papers showing their usefulness. From antibody design to multicellular function prediction, multi-omics, data integration, or predicting side effects. Yeah, so one particular aspect of graph neural nets here. So I'll explain a bit more about graph neural nets, but the idea is we're putting all of the information we have in this one graph, and you're going to have different types of nodes. What's exciting is that precisely work like the one that Zindlik, Agrawal, and Leskovec have done show that there's a huge potential for these kinds of techniques. We can use different types of nodes to represent different types of objects, and then different types of edges to represent the relationship between those objects. So in our case, we mostly have drugs and proteins as the main objects. So one way to think about this is there is the cell, and we are getting information about different parts of the story of what is happening as drugs are coming in and are having an impact through the nucleus and transcription on the behavior of the cell. And so the expression data gives us a kind of visibility on what is going on inside the cell. And we think that this is one of the crucial ingredients for the success of this project. Okay, so let me say a few words about graph neural nets. So the way that graph neural nets work is we have these nodes and edges in a graph, and some quantities are going to be attached to these nodes and edges. We can have both inputs and potentially targets in some places, so things we want to predict. Just like in normal neural nets, where this is like supervised learning, we have inputs and targets. But now the inputs and targets are attached to these nodes and edges. And then there's going to be some computation going on where the nodes are going to exchange information with their neighbors through some sort of messages. And then we're going to repeat that so that information can flow in the graph for many iterations. And eventually we stop that and we can read off with some output layers, some MLP, for example. In our case, will be on edges. We can have these predictions about the variables we care about, which here would be synergy. And of course, then we just backprop into the whole graph. And now it's a bit complicated, but it's something that's now well developed in the field. And there's a lot of code out there, and we're using PyTorch that can be adapted for the kinds of specific graphs that we care about. Now, let me say a few words about the uncertainty estimation and the acquisition function for active learning. In order to do exploration, as I said at the beginning, what we want is not just to think about what are our current best guesses, but also our uncertainty, because as you see in the figure, for example, if I have already these 3 data points, I could just go for the one that is best, but I already know the answer there, right? So it's not very interesting. I could explore around it, and maybe that's a simple way of searching. But I could also take into account the fact that there are areas in the input space, here this is molecular space, where I don't know enough, and maybe in those areas, for example, if you look at the mean minus the standard deviation, if we're going to minimize, we're trying to find energies, affinity energies, for example, or we're trying to find synergy, so maybe we're maximizing, sorry. So if you're maximizing here, maybe there's some configuration that is not as promising as the best one, but because of the uncertainty, there's a chance that it might be actually better. And so those more exploratory guesses we can use in this sequence of iterations where we send candidates to the assays. But for being able to do that, we need to estimate the uncertainty in our own predictions, right? So how do we do that? The classical way of doing this sort of active learning with Bayesian optimization relies on Bayesian integration in order to get an estimate of uncertainty, but that isn't always easy to do with deep learning. One of the methods that seems to work quite well and has been only recently really popularized is Monte Carlo dropout. So this is a technique for getting uncertainty around a neural net that has been trained by doing dropout, which randomly shuts off some of the units, and it's trained with dropout so that the model learns to be very robust to that noise that the dropout is creating at the places where there's data, because it wants to have the right answers there. But in places where there is no data, it doesn't care about producing such a confident answer. And so if we try to run the network with different seeds for the random generator that produce these random perturbations in the network, we're going to get different answers, and there will be more variability in those answers in places where the network, in a sense, has less certainty. Now, there is a more formal justification for Monte Carlo dropout, but it relies on the variational approximation of the Bayesian posterior. you can believe these approximations or not. And so in reality, what matters is that in practice, it seems to work fairly well. Okay, so then once we have an estimate of synergy as well as the uncertainty around it, how can we use this? So we could just not use it. This would be the greedy method. We could be even more agnostic and just take random pairs, or we could use one of the existing acquisition functions, or maybe even design our own. Two of the main ones we've been exploring is expected improvement and UCB. So expected improvement is interesting. It's saying, what's the expected gain over the best combination that I have found up to now. So we take the distribution over potential synergy. So now we're going to have a distribution because of our uncertainty. And we're only going to count, we're going to average how far we depart from the best, only for those cases where we do depart from the best. And so it's sort of an optimistic estimation of what we can expect that, that's better than what we have, the best we have found up to now. And then GPUCB is in a way much simpler. We're saying take the prediction plus a constant times our standard deviation, our estimate of uncertainty, and that's it, right? So kind of mu plus plus standard deviation with a scaling factor, and that actually works quite well, even though it's really, really simple. So let me show you some results. So first of all, we've done, again, these are experiments on the drug comb data set. We did experiments to see the importance of different input features. So higher is better here, and the x-axis is training epochs. And so what we see is that just looking at the protein target or the fingerprints could really be improved if we put in the gene expression. So gene expression, even alone, is a very strong signal, but gene expression plus fingerprint seems to be really the best. In terms of the different acquisition functions or using active learning or not, again, kind of repeating what I showed early on, we see that the use of these active learning methods, in particular the UCB method in those experiments, gives us a big gain. So in other words, as we do more and more assay experiments, so the x-axis here on the left is the number of calls to an oracle that is going to give us the answer for some chosen pairs. We make rapid progress in identifying much more rapid progress in identifying the best combinations. All right. Let me say a few words about next steps. So we've done these experiments on cancer data, and we've set up the pipeline. We're going to start the actual high throughput screening at the beginning of January. Our code is going to be open source. We still have a few engineering decisions to make on the model, but we are very confident that the pipeline will be right in shape for getting the new data at the beginning of the next year. Let me say a few words about some of the research questions that we are continuing to investigate. So one of the interesting questions with active learning is the fact that we can actually have multiple levels of fidelity from the assay. So you could imagine that you have assays that are more or less expensive to run and give us more or less accurate answer, or something that's more down the line. For example, in our case, we have these primary assays and secondary assays, and eventually mice assays. And of course, you might get less answers from the more expensive assays. Instead of thinking of just one oracle that gives us an answer, can we take into consideration the fact that there will be these several oracles, and the machine learning can decide not just what to send to the first line of assays, but also make choices about the second line and the third line and the fourth line. So this is a really exciting research area that we're working on. Another area that connects to our work on causality is to think about the graph neural net in a slightly different sense. So up to now, I've been talking to you about the graph neural net as just a way to represent all of the data we have. So that's one possibility. There's another possibility. We can think of the graph neural net to represent what is going on inside the cell, right? So we can think of it like a what if scenario simulator. Like, what if we take these two drugs and they interact with these proteins? What will be the outcome? How will it change what's going on inside the cell? So this is focusing very much on the question that I was alluding to earlier, of thinking about the pathways and the effect of the drugs on the activity of the cell. So then it would be a slightly different way of using the data, right? So we in a way, we have the general data set, but now for any particular imagined experiment where we consider, say, a pair or a triplet of drugs, we would run the graph neural net to simulate the outcomes that could be measured, like differential expression data. Right? So this is a very different way of thinking about the graph neural net, which we're working on. To close, let me step back a little bit and talk about causality, because a lot of this really is starting to poke at the question of the causal structure inside the cell. Now we can think of what we're really doing with those drugs is an intervention, like we're changing something in the way that the cell is operating. But we could do all other kinds of interventions, right? We could imagine we have extra nodes here which correspond to knocking off some genes, for example, right? When we start thinking about it this way, we are asking, what is the causal structure inside the cell? Of course, there's already a lot that is known by biologists, but there's also a lot that we don't know. Can machine learning help us figure out the missing parts, right? So let me share a little bit about, very briefly, some of the thinking that we and others, in particular in collaboration with the lab of Bernhard Schopkopf in Germany, are thinking about causality in its connection to machine learning and deep learning. And one of the fundamental ideas that we have started to explore in this respect is the idea that Schopkopf and collaborators explain in a 2012 paper of independent mechanisms or independent causal mechanisms. It's the idea that knowledge about how the world works, in our case, that might be just how a cell works, and a particular type of cell at that, that knowledge can be decomposed into pieces. And there is a way to decompose that into what's called informationally independent pieces. So in other words, it's not a statistical independence of the variables that those components operate on. It's an information theoretical independence, in the sense that, for example, if you look at the parameters that describe one piece, it doesn't teach you about the parameters of another piece, right? So if you had, for example, the same knowledge used in two places, like say two pink pieces of the puzzle here, and you didn't know that they really were, they should be copies of the same thing, there would be a strong dependency between what you learn for one of these pieces and what you learn for the other piece, right? And of course, what you'd like is to discover a knowledge representation that is able to figure out that there's only one pink piece, right? And it might be shared in different contexts. And why this matters is because when you do something to the system, which is called an intervention, normally what's going to happen is it's only going to touch one of these mechanisms. You're going to touch one of the variables, and maybe there's a drug that's going to activate a particular protein target. And of course, then there's going to be a chain of causal effects downstream. But to understand that the causal chain starts at this one place makes a big difference, and allows us to build learning algorithms that can find representations that have this property, that the changes that are happening are explained by a chain of causal effects that start in one place. And if you didn't factorize your knowledge in the right way, you wouldn't have that property. And what it also means is that it would take more data to learn. How much more time do I have? Sorry for asking.
There are 4 or 5 minutes.
Okay, perfect. I'm almost done. Right. So we have a lot of papers around this. Well, at least or ongoing or archive papers. And so some of our more theoretical work is about precisely this question of how do you discover the causal graph that relates a bunch of observations. And for now, we're only looking at the case where you can observe all the variables. The real prize, and this is also something we're working on, is where you might observe some of the variables and other variables you don't observe. And this would be the case in the cell, right? I mean, a lot of the things that are happening, you don't have any observation over. And so you'd like your model to discover these latent causal variables. So let me just mention a little bit about the team here. This is really a collaboration mainly between MILA and Relation Therapeutics. The project, the Recover project, is jointly led by Charlie Roberts at Relation and myself. And we have the support of David Roblin at Juvenescence, and at the Gates Foundation, Andrew Trister, as well as, of course, a number of folk on the ground at MILA and at Relation. Thank you very much.
Great. Thank you so much, Yoshua, for this fantastic talk. There are a number of questions, so let's start with those most popular. In case of safe polypharmacy, doctors often prescribe more than 2 drugs at the same time to patients. We attendees acknowledge this combination space for drug pairs is massive. So how can we hope to be able to model drug triplets or higher order combinations of drugs?
Right. Well, that's exactly why we are doing machine learning for this, right? The way to think about it is, as we acquire more information about the interactions between those drugs in one way or another, or even about individual drugs, right? Because like our graph neural net mostly has information about individual drugs and a little bit of information about some combinations. We are then able to evaluate in silico combinations that have never been tried, and then kind of optimize over choices of combinations according to the criteria we care about, which can include, of course, toxicity and other factors. So that's the idea, right? That we are able to do some of the search in silico, and of course, the problem is, how much data can we use to train these things? And you might think that there's not enough data, but the fact that we get a lot of information from the individual drugs makes a huge difference. So as you may have noticed in the experiments I reported, the number of pairs that was actually used is small. It's on the order of hundreds or thousands, and whereas the amount of data coming from the individual drugs, in particular, for example, the expression data we had was only for individual drugs, differential expression. Yeah, that's a way to deal with the lack of data here, that we have a lot of single drug information that can provide in a way very good features for predicting how pairs will behave.
Great. Next question. Expression profile of a specific cell type in which you test drug combination can impact the final outcome as well as affect that of the drug combination. Can you expand your ML pipeline to also recommend which cell line a particular drug combination should be tested on? And whether you are thinking about using public data on expression in specific cell types for that?
Yeah, that's a good question, and I'm not sure I have enough of the biology background to answer, but let me try. So, in our experiments, it's going to be easy, and we're going to have, we're not going to be making guesses like that. We're going to have the cell lines that we have, but in principle, in the case of the cancer data, you have a couple of thousand different cell lines. So. In principle, you can use the cell line as extra conditioning information. So what does that mean? It means that when you're making a prediction about a drug or a pair of drugs or a triplet of drugs, what you're saying, you're providing as extra input for the, say, answer in terms of inhibition, that you can provide as extra input information about the cell line. So then the question is, how do you represent cell lines? And one of the big questions in deep learning and machine learning applications is, how do you represent the inputs? This is a question that needs to be answered by talking to biologists. And what are the characteristics of cell lines that we can represent in a bunch of numbers that we could feed in as extra inputs? And that's the way I would deal with that.
Okay, thank you. There is a set of questions related to modeling and integrating messy data from a variety of experimental platforms that might come with their own artifacts and batch effects and biases. Do you have any models, and are you thinking about how to deal with this messy input?
The best thing is
and uncertainty in the graph structures and so on.
Right. So. Yeah, these are all hard questions, and I don't have the answers to all of them. But yeah, so graph neural nets, first of all, are like a good framework for incorporating many different types of data. So that's one thing that is nice. We can bring in studies that many different studies that were possibly made in different settings. Now, of course, this means if you don't have the right conditioning variables, then for the machine learning predictor, this is going to look like noise, right? And that's why I was saying it's important, if you can, to put in as extra inputs, even things like, in the case of batches, even knowing like what batch was it, like what day was it taken, and so on, can help. To learn, just maybe a few constants that can adjust for what's specific to that batch. So the general solution to this, if you can, is to provide extra input information that can help the learner see through the noise, some kind of regularity, which can help deal with that. But it's a hard question.
Great. Another question is related to your evaluation of active learning strategy in comparison to brute force approach. So comments from our colleagues from pharma industries, how is that different from what is done in pharmaceutical industry? No one does random brute force learning anyway. Can you comment? Do you have comment on that?
So what do they do?
That's a good question. And that's a magic. Good question. So
yeah, I mean, I'm quite happy that we could make other kinds of comparisons. So I imagine there could be all kinds of heuristics that could be used that would use the information available to help the exploration. And of course, there's a lot of work in statistics in trying to answer these kinds of questions. But here, I guess the difference is we are able to bring in these fairly powerful predictors that you wouldn't have in standard statistics, as well as estimators of what we call epistemic uncertainty. So the evaluation of uncertainty is actually a non-trivial thing. There's a difference that needs to be made between the uncertainty coming from the lack of knowledge and the uncertainty that is inherent in the data distribution. So the uncertainty that is inherent is because you're missing information, it's noise. Like even if you have infinite amount of data and you train the best possible model, your Bayes optimal predictor would still make mistakes, right? And would still have uncertainty. So that's aleatoric uncertainty. But the uncertainty we care about in active learning is the epistemic uncertainty. It's the residual uncertainty that you have because you have a finite amount of data, and that's the one that you're trying to get at. So that's I think this is a very current research right now to answer that question. I mentioned the Monte Carlo dropout, which is the most popular method these days, but I think there's a lot of exciting questions that can be asked to improve on top of that.
Great, thank you. I'll wrap up with a final question. Can machine learning help us find the right level of abstraction within biology? And I think the question here is referring to this notion of, should we do this modeling at the level of atoms and bonds between atoms, or whole proteins and protein interaction networks, or even going higher up in therapeutics level when we think of drugs and patients? Can machine learning help us model this?
I love that question. So let me remind you what deep learning is about. It should have been called representation learning. In fact, Jan Lequin and I, created a conference in 2013 called ICLR, International Conference on Learning Representations. And so it's all about discovering good representations. This is really what we're trying to achieve. And in the case of understanding how the cell works, you're right. I mean, it's not just the cell, actually. I'm thinking about these questions for climate modeling, for physical modeling of all kind. And what we have right now is scientists in a particular area over decades or hundreds of years, in the case of physics, have tried to come up with the right abstractions. So these are like aggregate variables. They're not at the level of atoms. They're higher up somehow. So think about temperature and humidity in the case of climate, right? So these are aggregate variables, and in biology, you might have expression data, you might have other sort of aggregate, like inhibition, that you're seeing in a cell is an aggregate variable. It's not at the atomic level. So we have these fairly obvious abstract variables, but really what we would like is to discover the right maybe missing abstractions that are going to help us capture the dynamics and the causal structure of what is going on in a physical system or in biology. And I'm not saying like we have a recipe for this. I'm saying the whole field of representation learning is really aiming at answering these kinds of questions. And I'm very hopeful that as we make progress, and especially with incorporation of causal reasoning and discovery, these are exactly the kinds of questions we're hopefully going to help deal with.
Great.
Basically, helping scientists to come up with the right abstractions for modeling phenomena.
Oh, that's a fantastic sentence to conclude the symposium. Thank you so much, Yoshua, for this visionary talk.